That's okay, chapter 14 is pretty short, so we'll finish 13 and do 14 on Monday. We've got an IRAD on Wednesday, and then we'll start the immune system and finish up the immune system on Wednesday of next week. Your next exam is on the 13th. Now, here's a kind of a kicker. I got an email. Did you guys get the email about?
Yes, SDSU letting their stuff.
Monday is no class of after April or after Easter. Yes, you got the email. So there's an email that went out. I will double check this, but there's an email that went out that the governor gave Monday after Easter as a holiday. It was never supposed to be a holiday. She's done this for the last couple of semesters now, and I need to start to anticipate this in the spring. She does it like 2 weeks before Easter, right? So you've got your whole things set and planned for your stuff, and then all of a sudden it's like, oh yeah, we're not gonna have class that day. And unfortunately, you guys are the ones that are affected, like, you know, it's the Monday class. So I will double check that. I'll make sure that we keep an eye on that. But that screws us up for the end of the semester because we're supposed to cover the whole respiratory system on the 18th. So that's a problem. So yeah, that's an issue. Anyway, we'll figure it out. So let's finish our, keep working on chapter 13 here. I think we got to this slide. We finished talking about this and the white blood cells. Did we start red blood cells?
Yeah, we ended with the hematocrit.
We talked hematocrit.
Hematocrit, yeah.
Hematocrit, okay. I gave you numbers for men and women?
Yeah.
For hematocrit, okay. So, remember that red blood cells, all blood cells are made in the bone marrow through a process called hemopoiesis or hematopoiesis. And in our red bone marrow, found in our skeleton, we make our red blood cells, our white blood cells, and our platelets. During development in the bone marrow, as the red blood cells are being produced and maturing, they spit their nucleus out. We've talked about this before in lab. They spit their nucleus out and then they get pinched in the middle. So when you look at red blood cells, they are very round and smooth and circular, and then they got this little indentation on both sides in the middle. And without a nucleus, you know, that sort of indicates that these cells aren't gonna live forever, right? You gotta have your brain in your cell. So red blood cells live about 120 days. They are destroyed in the liver and the spleen. They're destroyed by macrophages, some white blood cells that live in the liver and the spleen. And inside red blood cells, without a nucleus, the inside of the red blood cell is packed full of a protein called hemoglobin. And I don't have a picture of hemoglobin, but hemoglobin is a 4 subunit protein. So there's 4 separate proteins that come together to make up hemoglobin. So there's subunit 1, 2, 3, and 4. All of them together make up one hemoglobin protein, or hemoglobin unit. In the center of each of these subunits is an iron atom, and each iron atom can bind one oxygen molecule. And so each hemoglobin can carry up to 4 oxygen molecules. And in one red blood cell, there is a minimum of 250 million of these guys. 250 million. That's on the low end. That's a low end of the scale. So if each red blood cell, one red blood cell, has 250 million hemoglobin proteins, how much oxygen can one red blood cell carry?
250
Times 4, because each one of these can carry 4 oxygens, right? So 250 million times 4, anybody got it?
1 billion.
Yes. Each red blood cell can carry, at a minimum, 1 billion oxygen molecules. Billion oxygen molecules. And in our bloodstream, most of the blood is made up of red blood cells. 1 microliter of blood contains 5 million red blood cells. And nobody ever knows what a microliter is, but if you take a ballpoint pen and you push the tip up, that, what you have right there, sort of at the end of that, if that were hollow, that would be about a microliter. So in that much blood, there would be 5 million red blood cells. And if each of those 5 million red blood cells can carry 1 billion oxygen molecules, how much oxygen does the red blood cells carry?
So many.
Oh, a ton, right? Why? What's so important about oxygen? Why? Why? In what process? Thank you. To make ATP. Right. To make ATP. That's why we breathe. We breathe not just to give our lungs an activity, not just because we like the smell of air. We breathe so that our red blood cells pick up the oxygen from the atmosphere and carry it to every single cell in the body, so that those cells can take that oxygen, undergo cellular respiration, and make energy. Cellular respiration requires oxygen. If you don't breathe, what happens to you?
You die.
You die, because your cells aren't getting the oxygen and they can't make ATP and they can't keep their membranes together and they can't do their things and they die. And so do you.
So that drawing of the iron, is that for 1 red blood cell? Or is those individual?
This is 1 hemoglobin protein.
Okay.
1 hemoglobin protein, and there's 250 million of these in 1 red blood cell. Okay? Yes. This is 1 functional unit, and there's 250 million of these in 1 little cell.
Okay.
Okay. What happens if you don't have enough iron?
You bruise super easily.
Okay.
Anemia.
Because you have
Anemia.
iron deficient anemia. So if you don't have iron, guess what?
You can't carry much oxygen.
You can't carry as much oxygen, and what are you gonna have for low energy, lightheaded, pale, weak, easy bruising? That can also be associated with platelets, but
My mom is anemic, really, really bad anemic. And it messes with like everything.
iron deficient. Okay.
I just had to get hospitalized because of it. They told me to get hospitalized at a 2 and I was at a 4 on their test.
For what?
I don't remember what it was,
Mm.
but because it was
So normal hemoglobin levels for women range, I think it's 2 to 15. Men's is a little higher. I'm sorry, 12 to 15. Men's is a little bit higher. I think that's right. We're gonna do these. We're gonna do hemoglobin levels in lab. So we'll see what kind of hemoglobin levels we have. What happens if you just don't have enough hemoglobin? Same story. You're not going to carry as much oxygen. You have another form of anemia. Another form of anemia is called aplastic. Aplastic anemia means you don't make enough red blood cells. So if you don't make enough red blood cells, what are you not going to carry enough of?
Oxygen.
Oxygen. You're going to have the same story. Some form of anemia. And what's also going to happen to your hematocrit? If you have aplastic anemia.
Wouldn't it be lower?
What's your hematocrit? Percentage of red blood cells in a sample of centrifuged blood, right? So if you don't make enough red blood cells, what's gonna happen to your hematocrit? It's gonna be low, right? You have a low hematocrit. Instead of say 38 in women, maybe it's 28. Again, same story, you're gonna have low energy levels. Iron deficient anemia, what, do you think your hematocrit will be low? No, because you got plenty of red blood cells, you just don't have iron to fill them. You don't have iron to fill the oxygen, so your hematocrit typically won't be low in an iron deficient one. There's an anemia associated with low B12. Low B12 vitamin, you don't absorb it well. And then you don't make enough red blood cells. So again, your hematocrit will be low, energy levels will be low. And then there's sickle cell anemia, which we talked about, or we saw that picture in lab, where the red blood cells, instead of nice and round, they're more elongated. This is a genetic condition in which the hemoglobin subunits aren't nice and round, they're more elongated. And then that makes your hemoglobin elongated, which makes your cell elongated. And the hemoglobin doesn't bind oxygen as well when you have sickle cell anemia. So similar signs and symptoms, but different pathological process.
I read with the sickle cell that most people do not live as long.
Right. And it also depends on if you've got 2 bad genes versus 1. Right. You have different levels of badness. So when red blood cells are broken down by the liver and the spleen, the hemoglobin is also broken down, and one of the ultimate breakdown products of hemoglobin is called bilirubin. And if you've got issues with your liver, then the bilirubin is not excreted into the small intestine. So it gets excreted into the small intestine, where upon it is concentrated to make poop. And bilirubin is kind of a yellow color. And when it's concentrated, it is a brownish color, which gives poop its brown color. Then some of that bilirubin enters the bloodstream and heads to the kidneys, where it's not as concentrated, which is why urine is yellow. Poop is brown because it's very concentrated in the poop. And if your liver isn't getting rid of the bilirubin like it's supposed to because of cirrhosis, hepatitis, things like that, then that bilirubin goes directly into the bloodstream, and then what do you get?
Jaundice.
Jaundice. Yes, the bilirubin stays in the bloodstream, and you get jaundice. Your eyeballs turn yellow, and your skin turns yellow.
So what are Billy blankets doing to newborns?
So newborns, their hemoglobin is fetal hemoglobin. It's a little bit of a different structure than adult hemoglobin. And when they're born, that fetal hemoglobin will be replaced by the normal functioning adult hemoglobin. So the liver has to break down all that fetal hemoglobin, and sometimes the liver can't keep up. So newborn babies often have jaundice. Almost every newborn baby is going to go through a few days of turning a little bit yellow, while the liver is working overtime, dealing with all this extra sort of hemoglobin and that breakdown. The Billy blankets, what they do is it's a light source that breaks the bilirubin in the superficial vessels, capillaries and stuff, the capillaries that are close to the skin, and it breaks the bilirubin down so it's excreted better and more easily, because whatever doesn't get broken down in the skin goes right back to the liver and gets reconverted, and it's like a circle. So if the liver is overwhelmed anyway, what that does is it helps break it down and send it to the kidneys for excretion. So it works at the really superficial blood vessels. It's just an extra help. White blood cells, we're going to talk more about these in the immune system, so I'm not going to spend a ton of time on these. We have our white 5 types. We did this in lab. I haven't looked at those yet. I will look at those here in the next few days and post your scores on that. We'll talk more about it tomorrow. And remember, when you centrifuge the blood, most of the cells that get collected at the bottom are the red blood cells, and the white blood cells live only in this itty bitty little layer. Only 1% of all of these cells are white blood cells, and in that little layer there is your white blood cells and your platelets. So only about 1% of your cells are white blood cells, unless of course you have an infection. When you get an infection, then certain white blood cells that need to fight the infection get replicated, they get copied, and then your white blood cell count increases. Otherwise, and then after the infection's gone, those cells undergo apoptosis, and the numbers drop again to low levels. So we'll talk more about white blood cells when we get to the immune system. Platelets. Platelets are for clotting, and platelets are a piece of a large cell called a megakaryocyte. And the megakaryocyte is produced in the bone marrow just like all the other cells are. And then pieces of the megakaryocyte pinch off to form the platelets. So platelets are only a piece of a cell. And you get something like 2,000 to 3,000 platelets per one of these megakaryocytes. Resting platelets are nice and smooth. So you see over there on the left side, those are resting platelets. When the platelets get activated because of vessel injury, then notice they get spiky and sticky. And then we undergo clotting, which we'll talk about here in just a second. So resting platelets are smooth. When they get activated to help form a clot, they get nice and sticky and icky and help stick together. Hematopoiesis or hemopoiesis is the production of blood cells. Not just red blood cells, white blood cells, and platelets. And that occurs in your red bone marrow of your skeleton. Blood types. We're going to spend more time on blood types, again, in the immune system, but just sort of an introduction here. We got 4 blood types in the human population. Type A, B, AB, and O. Everybody know their blood type? Who doesn't know their blood type? Good, well you're going to learn it in lab. And if you don't remember, we'll confirm your blood type in lab. So, blood typing is all about red blood cells. It is a protein found on the surface of red blood cells only. These are not found on white blood cells, they're not found on kidney cells or skin cells or any other cell, they're only red blood cells. So if you are A blood, you have A-type proteins on the surface of your red blood cells. That makes sense. If you're B blood, you've got B-type proteins. And if you're AB, you've got both. If you're O blood, you ain't got either one. You're naked little red blood cells. Empty of A's and B's. So think of O as like 0. Now, the other thing that we have in our bloodstream are antibodies that we build automatically after birth. This will make a little more sense in the immune system. These are immune proteins that go off and fight infectious stuff, or foreign stuff, we should say. In your bloodstream, you also have antibodies that recognize the opposite blood type. And these are naturally occurring. They say they develop shortly after birth. It's not well understood how or why, but we do. So if you're A blood, you've got B antibodies. If you're B blood, you got A antibodies. And if you're AB, you don't have E either, because you wouldn't want antibodies attacking your own blood cells, right? So you can't have either. You can't have any antibodies. And if you're O, you have actually both. So if I'm A blood and somebody were to accidentally give me B blood, then my antibodies in my bloodstream would bind to the B blood cells and target it for destruction. And if somebody gives me a transfusion of B blood and I'm A blood, the antibodies that are found in the transfusion would cross-react with my red blood cells and blow them up as well, or target them for destruction. That's a bad thing. You don't want to do that. You don't want to screw up the blood typing and give somebody the wrong blood type. So you want your own blood type. The other thing that we have on blood cells, and we'll go over this a little bit again in the future, so don't get too tied up about it right now. The other thing that we have on our red blood cells is we either have it or we don't, the Rh factor. And the Rh factor comes from the word rhesus, for rhesus monkey, because it was discovered in the rhesus monkey. So if you're AB positive, you have A's and B's and RHs on the surface of your red blood cells. If you're O negative, you got nothing. If you're A positive, you've got the A's and the RH. Let's say if you're B negative, you've got the B's but no RH. And we don't have anything to do with these naturally occurring antibodies with regard to the RH. We don't have those. If you're negative, like O type, O negative has neither A or B, but they've got both antibodies, but they don't have RH. You develop that later if you experience it, or if you see it. So we'll talk more about that later.
Why,
So, yep.
Why do you have, like, why is, like, most people have, like, A positive?
85% of the population is Rh positive. 85% of the population is Rh positive. That means 15% is Rh negative. So fewer people are Rh negative than are Rh positive. It's just part of our genetics. And then depending on ethnicity, different blood types are prevalent with different ethnicities.
What about O positive?
What about it?
Does it have RH?
Sure.
Okay.
Most Caucasians are Rh positive, are O positive, sorry, O positive. Most rare blood type is AB negative. The most rare blood type.
And it's just because of genetics that occur.
So what happens when mom is negative and
We'll talk about that later.
Okay.
Hang on to that question. We'll do it. We'll do it. Most rare blood type is AB negative. All right, clotting. Clotting has to do with our platelets. Platelets are for clotting. When you have a vascular damage, when you cut yourself, we have to stop the blood flow. And platelets are a very important aspect of doing this. So let's look at this little picture. Here we have a blood vessel that's been sliced open. The first thing that happens is the damaged cells are going to release chemicals. These chemicals are known as something called a cytokine. A cytokine is just a chemical released by a cell, and it causes an action in another cell. Cytokine is like an umbrella term. Hormones could be considered a cytokine, right? Because they're released by specific cells, they go to another cell and they cause an action. They happen to go through the bloodstream. Cytokines can also go through the bloodstream. The immune cells release tons of different cytokines, tons. And what they do is they go to different locations in the body and they cause an action. So all of these cytokines have different names. We're not going to talk about some of these different names, but like one of them is called IL-1, and there's tumor necrosis factor, and there's other ones that are out there. But when you have damaged cells, when cells die by necrosis because of damage, those damaged cells are going to start releasing these chemicals in the area, and that chemical is going to diffuse. What that chemical, that cytokine, that form of a cytokine does is it causes the vessels to vasoconstrict. It causes the smooth muscles to squeeze. So if they squeeze, that slows the blood flow, right? We got to stop the blood flow. Those cytokines also activate the platelets. And so then the platelets come in that are flowing through the bloodstream, and they swim through the cytokine chemical, and then they get spiky and sticky. And what they do is they stick to the exposed collagen of the blood vessel, and they stick, and they all stick together. When they get activated, and they get spiky, and they get sticky, and they start sticking to that blood vessel, they release more different types of cytokines. And those cytokines activate our clotting factors. Clotting factors are proteins that travel through the bloodstream individually, all happy, doing nothing unless they are swimming through the cytokines from the platelets. So now we've got these platelets that are releasing cytokines, okay? Clotting factors, there's 13 different clotting factors. 13 clotting factors are 13 different proteins floating through the bloodstream. And when they come through and they swim through the chemicals from the platelets, then they start sticking together as well. And clotting factor 1 will come in, and 2 will come in, and 3 will come in, and 4 will come in, and 5, 6, 7, 8, 9, 10, 11, 12, 13. And what they do is they form a spiderwebby mesh called fibrin. And that fibrin is literally like a spider web, right? You ever walk through a spider web? It's sticky and thin and gross. Not that fibrin's gross. But what that does is that spiderwebby network makes more platelets stick, and even red blood cells stick. And so now you've got this sticky spiderwebby mesh with platelets and red blood cells, and that sits over your injury, and that is a clot. And after all those 13 clotting factors come in and they form that spiderwebby mesh, then what happens is that mesh squeezes and pulls the edges of the injured vessel together, and now you have a clot. The other thing that is included in this spiderwebby mesh is a protein called plasmin. And what plasmin does is, as that vessel is repairing itself, as that vessel is repaired and closed over, we got to get rid of this clot, right? And we can't just say the clot just can't release and go floating through the bloodstream, because then what would happen? You get a blood clot, right? You get a blood clot in your brain, you get a blood clot in your lungs, so we can't do that. So what happens is plasmin, after the vessel is healed and closed over, plasmin dissolves the clot into itty bitty little molecular components, so we don't have a great big pile flowing through the bloodstream, heading to the brain and causing a stroke. So it's a really complicated process, but what part of the body isn't, right? What happens if, so we said there's 13 clotting factors. What happens if you don't have number 8?